Hello dear students, welcome in this lecture series on genes and chromosome. I am Professor Hemlata Suryawanshi, Assistant Professor in the Department of Microbiology from Pratap College, Amalner. Today we are going to discuss about plasmid, its concept, types, structure and properties as well as incompatibility. So let's discuss about concept of plasmid. You can see this image of bacterial cell. This bacterial cell possesses main chromosome, bacterial DNA, which carries different types of functional genes. And also you can observe such a small circular part. So these are extra chromosomal entities which are small, circular, having self-replication ability. They are double-stranded DNA molecules and independently they exist into the cell beside this main bacterial chromosome, and they are known as a plasmid. So plasmid is nothing but plasmids are nothing but small circular self-replicating double-stranded DNA molecule that exist independently of a chromosomal DNA that we can define. The term plasmid was introduced by Joshua Lederberg. The credit goes to Joshua Lederberg. So in the year of 1952, uh, he was introduced plasmid to us. Where are plasmid found? See, plasmids are mainly observed, mainly present in bacteria. So, in bacteria, the role of plasmid is to transfer antibiotic resistant characteristic from one cell to another cell. They also uh, having ability they also having ability to transfer these genes through conjugation, formation of the conjugation bridge, and because of presence of such a types of plasmids, they can adapt quickly the environmental stress condition. Also these plasmids are present in archaea. As we know that archaea are that types of bacteria which can withstand the extreme conditions. So this ability to withstand, suppose, highest temperature, then high salt concentration, this ability or these characteristics are present in archaea due to presence of such a types of special plasmids. Plasmids are also present in some yeast. For example, 2-micron plasmid size containing Saccharomyces cerevisiae. Plasmids are rarely present in higher eukaryotes. As you know that plants and animals, they do not possess the plasmids. So these plasmids, we can say that scientists, they artificially introduce these plasmids into the plants or animal cells during genetic engineering experiments. Now let us see the structural feature of plasmid. Plasmids are circular, they are double-stranded DNA, and size of plasmid ranges from 1 kilobase up to 200 kilobase. Plasmids are mostly present in the supercoiled conformation. So what is the meaning of supercoiled conformation? Here, the DNA get twisted upon itself. That is known as a supercoiling. So this supercoiling makes the plasmid most compact and allows it to fit easily inside the cell. Now, in this image, you can see that various regions on the plasmid. This is the origin of replication. This is the small segment of the DNA. From this, actually replication starts. Then it also possesses the selectable marker genes. So these marker genes enables to identify the particular plasmids. Suppose ampicillin resistant plasmids are there. If we grow organism into the media which contain ampicillin, if organism shows growth in presence of antibiotic, then we can say that these organism possesses ampicillin resistant gene. So these are such a types of selectable marker genes which are present in the organism. Certain regulatory genes are present there. So these regulatory genes, they control the replication also, as well as copy numbers and gene expression. Sometimes transfer genes are also present. So these transfer genes which are responsible for conjugation. There is a conjugation takes place in between F plus cell and F minus cell. So for that purpose, for transferring the resistant antibiotic resistant genes or other characteristic from one organism to another organism, such a transfer genes are present on the plasmids. Now, based on function of plasmids, see, there are different types of genes which are present. They carried out various types of function. So based on that functions, we can classify the plasmids as a F plasmid, which we call them as a fertility plasmid. So these plasmids, they carries the gene for conjugation. Actually, F plasmid possesses transfer genes, TRA genes. Okay, so that bacterial cell which possess the F plasmids are known as a F plus cell, and those bacterial cell which do not possess that F plasmids are known as a F negative cell. You can see in this image, this is the F plus cell and this is F minus cell. So what happens? F plus cell, they are having ability to form the bridge in between F plus and F minus cell. So the sex pillars which present on F plus cell, they elongate and to form the bridge to conjugate in between F plus and F minus cell. So through this bridge, as we know that plasmid is a double stranded, so one of the strand, it enters into the another plasmid. So the characteristics possessed by a plus plasmid, now these characteristics are also shifted through the transferred through the plasmid genes into the F minus plasmid, and now F minus cell is also become F plus cell, and that is also now become donor cell. So this type of conjugation we observed in Escherichia coli. In E. coli, we can observe such a type of conjugation. Okay, so that is also known as a F factor plasmid, as the transfer genes are present in them. So now, next type of plasmid is a R plasmid. R plasmid is a resistance plasmid. So such a type of plasmid carries the genes which shows the resistance towards antibiotic or poisons. For example, resistance to tetracycline antibiotic, ampicillin antibiotic, as well as at the same time some of the bacterial cells showing resistance towards the multiple drugs. So multi-drug resistance is also the important characteristics of some cells which possesses R plasmids. Coal plasmids are also there. The next type, that is known as a colicin plasmid, as there is one of the protein which is the toxin known as a bacteriocin, produced by some organisms which possesses coal plasmids. Such a type of bacteriocin, they are having it is having ability to kill the other bacteria which showing the competition with the organisms for nutrition or for shelter, etc. So that are nothing but the proteins. These plasmids or bacteriocins are nothing but the proteins which possesses by E. coli and which is encoded as a col E1 plasmid. Now, another type based on function is a virulence plasmid. So, such a type of organisms, such a type which possess the virulence plasmid, they carries pathogenicity genes, means that organism possesses disease causing ability. For example, tumor inducing plasmids, which are present in the Agrobacterium tumefaciens, which are known as a TI plasmids. So, as they are pathogenic organisms because of presence of such a virulence plasmid. Degradative plasmids are also there, which helps in degradation of the unusual substances, or simply we can say metabolism of unusual compounds. For example, TOL type of plasmids which are present in the Pseudomonas putida. Cryptic plasmids are also there. So that of the cryptic plasmids, their function is unknown. They do not carry any type of useful genes. They still replicate, but their function is not clearly understood. Here you can see different types of plasmid and their function. So shortly, we can say that F type of plasmids, they carried out conjugation. R type of plasmid, this plasmid possesses antibiotic resistant gene, and the organism which possess the R plasmids, they are showing resistant towards particular antibiotics. Coal plasmids are there. The organism which possess coal plasmid, they are having ability to produce bacteriocin. Then virulence plasmids, if it present in the organism, then the organisms are pathogenic in nature. They can cause the or having the ability to cause disease. Degradative plasmids, if present in the organism, then the organisms can metabolize certain unusual compounds. So in this way, there are different types of plasmids and their functions. Now we move towards the properties of plasmids. See one by one. One of the property of plasmid is autonomous replication, self-replication. Plasmids do not depend on chromosomal DNA in the bacterial cell for replication. They independently replicate. Then copy number. Some of the plasmids are present in number 1 or 2 per cell. So they are known as a low copy number plasmid. Some plasmids are from 10 to 100 copies per cell. They are known as a high copy number plasmid. Their size is also variable from 1 kilobase to 200 kilobase. Then transferability. Conjugative plasmids, they are having this ability to transfer plasmid genes from one organism to another organism. Non-conjugative plasmids are also there. Then host range. Plasmids, some of the plasmids, they showing narrow host range, only certain type of host they are present, whereas some of the plasmids are almostly present in many of the host, means they are possessing the broad host range. Curing of plasmid, this is also one of the important properties. Here, due to certain factors like heat, UV radiation, then acridine orange, these types of factors are responsible to remove the plasmids from the cell. So plasmids are also susceptible for towards heat, UV radiation and acridine orange. These are the properties of plasmid. Now we move towards one of the major concept, that is incompatibility of plasmid. Plasmid incompatibility, it is refers to inability of two different plasmids to stably coexist in same bacterial cell over many generations. So if two plasmids which are present in the same cell from many time period, from many generation, then one of them will eventually be lost during cell division. So we can say that these two plasmids are incompatible plasmid, and after few generation, one copy, if the two plasmids are same, then one copy get lost during the cell division. Then what is actually the mechanism behind this incompatibility? See, replication of the plasmid, which is strictly controlled by replication initiation proteins, antisense DNA regulators, and also the copy number control system. So there should be the control on copy numbers. If two plasmid share the same regulatory system, then what happens? The cell treats them as identical copies of the same plasmids. And during cell division, some of the daughter cells may receive only one plasmid type, not a both. And over time, if the prolonged time period they are coexist in the same cell, then one plasmid is lost from the population. So this happens in case of the incompatibility of plasmids. So students, in our next lecture, we will discuss about genetic code and its properties. So till then, goodbye. See you in the next lecture.